Good morning, everyone. Today topic is Agrobacterium. Agrobacterium. So in this topic, we discuss two important Agrobacterium species. Yes. So generally, Agrobacteriums are gram negative bacteria. Gram negative bacteria, and they are soil-borne bacteria. Soil-borne bacterium. And generally, this bacteria has this is the structure of bacteria. In this bacteria, we have two important structures. Like this bacteria contain their own genetic material, like this, and also this bacteria contain extra chromosomal genetic material, like this. Like this bacteria contain two different types of genetic material, and this the main genome of this bacteria, main genome, are also called bacterial chromosome. Bacterial chromosome are also called, the main genome of this bacteria is called genophore. Genophore. And along with this main genome, there is a extra chromosomal and circular double-stranded self-replicating genome is present, and this genome is called plasmid. Plasmid. Generally, this plasmid has the nature of circular extra chromosomal. It is extra chromosomal, and it has double-stranded DNA, and it has the nature of self-replicating. Means it doesn't depend any other structure. Self-replicating genome. Self-replicating genome it has. Here, important note here. Generally, mostly gram-negative bacteria are pathogenic in nature. Pathogenic in nature compared to gram-positive bacteria. Generally, gram-negative bacteria are pathogenic in nature. Means they cause disease to the various organisms. Here we will go through. We have two different types of Agrobacterium species. Yes. So in Agrobacterium genus, in Agrobacterium genus, we have two different types of species. Two different types of species. So they are Agrobacterium tumefaciens, tumefaciens, and one more species in Agrobacterium, that is Agrobacterium rhizogenes. Rhizogenes. So likewise, we have two species in Agrobacterium genus. Now we will discuss each species in their importance. Generally, this Agrobacterium tumefaciens which cause disease. Which cause disease, that is Crown Gall disease in dicotyledon plants only. Very important. It causes disease only dicotyledon plant. And this Agrobacterium rhizogenesis, it causes disease called hairy root disease. Hairy root disease. And this Agrobacterium tumefaciens species which contain a plasmid. called TI plasmid. TI plasmid. Like this, for example, it is a structure of Agrobacterium tumefaciens, and it has its own genetic material, genome or bacterial chromosome, and here we have plasmid like this. This plasmid is called TI plasmid. TI plasmid. So TI plasmid present in Agrobacterium tumefaciens. Here TI means tumor inducing plasmid. Here I am writing tumor inducing plasmid. So like TI plasmid, in Agrobacterium rhizogenesis also it has a plasmid called RI plasmid. RI plasmid. Here RI means root induced plasmid. Root inducing plasmid. Like this is the Agrobacterium rhizogenesis, and this bacteria also has its own genetic material, and here extra circular double stranded genome, that is RI plasmid. RI plasmid. And now these TI plasmids. This TI plasmid contain tDNA, transferred DNA, contain tDNA, and this RI plasmid also contain a DNA called rDNA or DNA, and these tDNA very important. This tDNA contains two important genes. They are weed gene or weed gene operon, virulence gene operon it contains, and also it contains auxin, very important, auxin and cytokinin gene. And these genes like weed genes, auxin and cytokinin genes all are oncogenic genes. Oncogenic genes. And these genes which cause disease, that is Crown Gall disease. Crown Gall disease in dicotyledon plants. How many they contain? Like this, for example. Yeah, here. For example, it is a TI plasmid. In this part here we have tDNA. tDNA. And this tDNA contain, in which tDNA contain both weed gene operon and auxin and cytokinin gene. And these genes act as a oncogenic in nature, and this due to presence of this gene it causes disease to the dicotyledon plant, that is Crown Gall disease. Now coming to rDNA. Here this, for example, here we have RI plasmid, and this RI plasmid contain small portion of RI plasmid contain a DNA called rDNA. So this rDNA which contain a gene called, this rDNA contain a gene called role gene, ROL, role gene. And now this gene is a oncogenic gene, and which cause disease, that is hairy root disease. Hairy root disease. The important thing here in this lecture, and if we use this tDNA, if we disarm, very important concept here. If we disarm, here I am writing in another slide. Yes. If we disarm, here note, if we disarm the pathogenic nature of these oncogenes in tDNA as well as rDNA, then the genes, we can use those genes as a cloning vector in plant. We use them as a cloning vector in plant. How means? Generally, for example, here, this is what TI plasmid or RI plasmid thing, and in this we have this tDNA portion, and this tDNA contains auxin and cytokinin gene, and also it contains Vir gene opera. Vir gene opera. If you disarm this part, tDNA, if you disarm this part, disarm this tDNA. Here disarm means the genes causing the genes causing tumor growth like oncogenes we need to them remove or we can modify them modified. So here we need to remove or modify the tumor growth genes like oncogenes. Like here oncogenes are auxin and cytokinin genes, and here in RI plasmid we can roll genes. If we remove this oncogenes or tumor growth genes, and remember this roll gene in and Agrobacterium rhizogenesis only. I am talking about tDNA related. So hence here we can remove this auxin and cytokinin gene from tDNA, and then we can use them as a cloning vector in plants. Hence this Agrobacterium tumefaciens is also known as natural genetic engineering. Very, very important. Hence we can use them as a natural genetic engineer. So which microbes we can use them as a natural genetic engineering means to Agrobacterium tumefaciens, because here we are disarm the tDNA, means we can remove the pathogenicity of these oncogenes like tumor growth genes, auxin and cytokinin. Then we can use them as a cloning vector. Hence we can call them as a natural genetic engineers. So this is all about the concept of Agrobacterium genus species. In Agrobacterium, we have two important species like Agrobacterium tumefaciens and Agrobacterium rhizogenes. So this is all about the Agrobacterium. So here we have three highly important NCERT based MCQs I have provided, and read these three MCQs. Yeah. So here asking about Agrobacterium tumefaciens. Agrobacterium tumefaciens species is widely used in plant biotechnology. Yes, it is widely used in plant biotechnology as a cloning vector. Just now we discussed why. It has the natural ability. Means read this four and choose the right answer, and those answer must and should be drop in comment box. One more MCQs. Yeah. Yes. So here, second one, which of the following statement is correctly regarding Agrobacterium? Here also asking about Agrobacterium tumefaciens. Here. And often referred to as a natural genetic engineer. Yes, just now we discussed why Agrobacterium tumefaciens are referred as a natural genetic engineer to the plant only. So here there are four options. Read it. And one more MCQs. And it is the last MCQs from this topic. Yeah, here we have. Yes. So here the third MCQ is asking about another species in Agrobacterium, that is Agrobacterium rhizogenesis. Agrobacterium rhizogenesis is widely utilized in plant biotechnology. Yes, for hairy root culture because it contains. So read this four options and drop your answer in comment box. So thank you. Have a nice day.